Hello, everyone. I'm Cailin Spencer,
I'm Gordon Graham.
And I'm Kyle Byron.
and thank you for tuning in to our presentation on the effects of Wi-Fi exposure on the ATU1 gene in the model organism Tetrahymena thermophila.
All right, so for a little bit of introduction to our presentation, we looked at Tetrahymena thermophila as the model organism for our study because it is a eukaryotic single-celled organism with a genome very similar to the human genome and has high proliferation rates. So it's very useful for modeling cell behavior. So for the background to this project, we looked at the effects of Wi-Fi radiation originally in a Denmark study about the effects of Wi-Fi on plants, but this was then expanded to studies done in 2016 that looked at the effects of Wi-Fi radiation on rats. Specifically how Wi-Fi routers have a detrimental impact on the reproductive system of rats and of anything with sperm cells. So using this, we looked at alpha tubulin, which is a ubiquitously expressed protein that plays a role in sperm motility, and it also plays a similar role in Tetrahymena thermophila, the motility of the individual cells themselves. So for our hypothesis was that if we expose Tetrahymena to the EMF radiation, then the growth and feeding of the cells will be reduced and the expression of the ATU1 gene will also decrease.
Okay, so to conduct this experiment, we stationed 2 samples of Tetrahymena that we cultured in between a 2.45 GHz Wi-Fi router and a sample computer connected to that router. So data was sent back and forth between the computer and the router for 72 hours with our samples right in between them. Every 24 hours, we performed a cell count on our samples using the hemocytometer, and at the end of the 72 hours, a feeding assay was performed using India ink. So to quantify our ATU1 gene, we first created a primer for that gene using the oligoanalyzer software. And then we performed an RNA extraction and reverse transcribed that RNA into cDNA using RevertAid. And then that cDNA was quantified using PCR reaction.
So for our results, we had a series of 3 graphs, one of which was taken from the cell count that we did from the hemocytometer. The next, also using the microscope slides of each culture, we examined the feeding vacuoles of the Tetrahymena. And finally, using our qPCR reaction, we looked at the fold behavior of the gene, the ATU1 gene, between the control and experimental studies. So for the first figure, you see the number of cells per milliliter of media, which was averaged between both cultures in the control and in the experimental groups. At 24 hours, we had a p-value of 0.283. At 48 hours, the p-value was 0.391, and at 72 hours, the p-value was 0.652, and we had 3 replicates for each group. Moving on to figure 2, looking at the average number of feeding vacuoles under each slide, we had a p-value of 0.807 with 30 replicates per group between the 2 control and experimental averages. And then for figure 3, this is looking at the fold change expression of the ATU1 gene. So in the control, we have the expression set at 1, and then in the Wi-Fi exposure experimental group, the expression rate went down to 0.0184 with 2 replicates for each group.
So in conclusion, our cell counts and our feeding assays indicated no statistically significant difference between the control and experimental cultures, and those p-values can be seen in figures 1 and 2. And the single round of quantitative PCR is inconclusive. The expression of the ATU1 gene may or may not be significant. More rounds of qPCR will be required to extrapolate a p-value, and then determine the significance of any difference observed. So our results suggest that EMF-RF radiation emitted from 2.4 gigahertz Wi-Fi routers do not damage alpha tubulin structures or the ATU1 gene in Tetrahymena Thermophila, and these results do disagree with our hypothesis. Future experiments should probably include more rounds of qPCR, like I said, to improve the integrity of the data and get an actual p-value to determine the significance of any differences that are observed. And we also think that it would be useful to include a computerized motility assay. Tetrahymena Thermophila rely heavily on the alpha tubulin structures in their cilia to move, so data collected from their motility would be useful in determining whether or not the alpha tubulin structures, even if they are expressed, are damaged or not. We would like to thank our faculty advisor, Dr. Otto Hitt, for her guidance and support throughout the duration of this project. And we would also like to thank Ryan Klingeman with the Carroll College IT Center for providing us with the wireless AP and helping us set up all the computer-related stuff for this project. So thank you to you guys. We would also like to thank everyone tuning in for taking the time out of their Friday to listen to our research that we've done this semester, and we hope that you have a great rest of your day. Thanks.
Thanks.